KURINJI Senior Secondary School, CBSE, Namakkal.
Successful result producing school in state every year.
Administered by the Board of Directors with over 35 years of experience in the teaching field.
Special attention on art, craft, music, dance, silambam, yoga, skating and karate.
Focus on Hindi Sabha and Spell Bee Olympiad exams.
Smart class for effective and innovative learning and well-furnished library.
Solitary importance on sports and games in state and national level.
WhatsApp facilities to know activities of students and school inside the campus.
NEET, JEE, IIT coaching experts from Rajasthan and Andhra Pradesh along with our effective teachers.
Admission registration for classes LKG to +2.
KURINJI Educational Institution, Kavetipatti, Namakkal. Admission in progress. CBSE, KG to 12, contact 9025895176. Metric 6 to 12, contact 9344567484. For online admission, log on to www.kurinjischoolnkl.in.
Students, in this video we are discussing remaining information of earlier experiments. So in a photosynthesis introduction point of view, we have learned Joseph Priestley information and John Ingenhouse's information in previous video. Any earlier experiments concerned, so and a few more important points of or experiments of remaining scientist information. Now let us see. So it is video number 2 under photosynthesis in higher plants chapter. Now Julius von Sachs. Did you remember this person name? So Julius von Sachs. So in a mineral nutrition chapter, we have seen the demonstration of hydroponics for the first time done by the Julius von Sachs only. Okay, Julius von Sachs provided evidence for the production of glucose when the plants grow. Okay, so means here, so what is the information? For the first time the plants prepares the food materials so by the process of photosynthesis, and later what he was given evidence means, so production of glucose means so here Julius von Sachs for the first time he was given or he was provided evidence for the production of glucose by the plants. Okay, later glucose was usually stored as a starch. So generally glucose, how it will be stored means it stores as starch. So means for example we will have a potato now, so in the potato stored food materials are starch. Next one, his studies showed that the green substances in plants means chlorophyll is located in the specialized bodies later called as chlorophyll within the plants. Means here what was given, so there in plants, so specialized green color pigment or the green color substance will be there. So that particular green color substances only nowadays we are calling as chlorophyll, and this particular chlorophyll which is present in this specialized structure known as chloroplast, and the entire cell or entire chloroplast will be there in the plant cells. Means so thereby what is the information in the cell unit of life chapter we have discussed the structure of chloroplast. In the structure of chloroplast we see structure of chloroplast, within that thylakoids are present on the thylakoid membranes. On the thylakoid membranes we have seen the presence of chlorophyll pigments which absorbs the light. Okay, so points we see now, so that is a point he was given by those points were given by the Julius von Sachs. Later he found that the green parts in the plants is very so green parts of the plants, so these parts where the glucose is made. So again he given or he found that one, so in the green plants or in green parts of the plants he found or he explained that the glucose synthesis or the glucose where the process is going to be taken place, he found it, and that glucose is usually stored as a starch again. So same point he was repeated to give. So thereby here one point mainly we can understand, so related to the Julius von Sachs. For the first time he was provided one evidence so here for the production of glucose by the plants. So there also when we are going to deeply to know about these particular studies now, So their green color pigments are present. These green color pigments only nowadays we are calling as chlorophyll pigments. Now these chlorophyll pigments are present in the special bodies known as chloroplast, and these special bodies are present within the plant cells. Means here if you see there no plant cells are present, and within the plant cells chloroplast cell organelles are present, and within the chloroplast cell organelles we are finding the presence of green color substances. So likewise here on a flow chart information we are discussing related to the structure of chloroplast as well as the function of chlorophyll pigments. So later, so definitely by the process of photosynthesis glucose is going to be prepared, and the glucose generally which is stored in the form of starch. So this entire information was given by Julius von Sachs. Later T.W. Engelmann. T.W. Engelmann is a fourth scientist who gave the information related to the process of photosynthesis. So what he was done in 1843 to in 1901. So using a prism. So in a physics lab, so generally we will notice the prism. So by using of prism, he split the light into its special components and then illuminated a green alga chlorophylla. So thereby here T.W. Engelmann, so by using the help of prism, he was succeeded in separation of light into 7 parts. That is only we are calling as VIBG or no. So that one, and later what he was done in his experiments. So he was used the one important bacteria, one important algal member name is Cladophora. Okay, so here the Cladophora was used, the Cladophora green algae was used to his experiments, and the Cladophora bacteria when it was placed in the suspension of it comes under aerobic bacteria, and the bacteria were used to detect the sites of oxygen evolution. So means here already I told you once oxygen evolution process. So by the splitting of water with the involvement of manganese, so here water is evolved, likewise we seen. So thereby here, so for this experiments point of view, so Engelmann was used the green alga name is Cladophora. Next one, he observed that the bacteria accumulated mainly in the region of blue and red light of the split spectrum. So thereby one more part what it was used. So when by using of this prism, when he was divided the light into different parts, again he was observed the more accumulation of the green algae. So he was observed more accumulation of the green algae was observed in the red light and blue light. So thereby here this particular information what it was giving. So the region of blue light and red light of the spectra, you found more number of Cladophora. Okay, next one, a first action spectrum of photosynthesis was thus described. So thereby here, when he was given information about that, he was succeeded in giving of first action spectrum of photosynthesis. Later, it resembles roughly the absorption spectrum of chlorophyll A and chlorophyll B. So means here what is the information he was given perfectly, those that particular information it is 100% related to the absorption spectra of chlorophyll A and chlorophyll B. Okay, absorption spectrum of chlorophyll A and chlorophyll B. Later by the middle of the 19th century, the key features of the plant photosynthesis were known. So means by the 19th century, so our scientists they have found out, they observed one particular key point, that is so namely that those plants could be use the light energy to make the carbohydrates from the carbon dioxide and water. So it means in mid 19th century or in the middle of the 19th century, so what we were found means we succeeded to found that definitely plants has the capability to prepare its own food with the help of light energy, with the involvement of light energy, and with the help of carbon dioxide and water, plants can prepare the carbohydrates. So likewise, so one important points or key features we have learned or we have studied or we have observed in the middle of the 19th century. So now here, what is the point here? Once you see, first equation of the photosynthesis here is carbon dioxide, water, and with the presence of light, so CH2O, later oxygen. So here this particular reaction they have given. So here CH2O for the first time they have been represented. So the this chemical compound, it is first represented as carbohydrate. Okay, so that is represented as a carbohydrate. Later, what are the points that we have discussed now? So this particular carbohydrate is nothing but glucose. It is a 6th carbon of sugar. Okay, so it is a glucose, it is a 6th carbon of sugar. Likewise, so for the first time they have given information regarding the formation of carbohydrates. For the first time they have been noticed initially. Later, a milestone contribution to the understanding of photosynthesis was that made by the microbiologist, and the microbiologist name is Cornelius von Nile. One more time, the milestone contribution to understand the photosynthesis was that made by the microbiologist Cornelius von Nile. So NIEL, Cornelius von Nile, who based on his studies of purple and green bacteria. Okay, so means he was studied on the which one, purple and green bacteria. So later what he was given information demonstrated that the photosynthesis is essentially light dependent reaction. So means here he is giving information, continue to give the information regarding the photosynthesis. So what he was given, so by using of purple and green bacteria, he demonstrated that photosynthesis process is a essentially light dependent reaction in which hydrogen from a suitable oxidizable compound reduces carbon dioxide to carbohydrates. So means here again continue to give us this information. So according to this information, what is the information he is giving us? So there oxygen definitely is going to be released, and so from the carbon dioxide to carbohydrates formation, and this entire carbohydrates process formation definitely is a light dependent reaction he was given. So based on this one, what is the point? This can be expressed by the, once you see there, so 2H2A plus CO2 in the presence of light, 2A plus CH2O plus H2O. So likewise, and the second reaction, so reaction or the chemical reaction that were given by the or demonstrated by the, so which one, microbiologist Cornelius von Nile. So by observation or by studying of purple and green bacteria. Okay, so later in green plants, okay, in green plants, what is information? H2O is the hydrogen donor. In green plants, H2O is a hydrogen donor and is oxidized to O2. So it is the hydrogen donor. Hydrogen donor means water, which is going to be oxidized to O2. Next one, some organisms do not release O2 during photosynthesis. So means here green plants, if you take there, no, so these green plants by using of water, water is a hydrogen donor and is oxidized to O2. Next one, in some organisms do not release O2 during photosynthesis. When H2S, instead is the hydrogen donor for purple and green sulfur bacteria. The oxidized product is sulfur or sulfate depending on the organism and not O2. So thereby here what is the point? Suppose if you take some other kind of organisms, so here what is the point? Hydrogen donor for H2S, hydrogen donor for purple and green sulfur bacteria, hydrogen donor is H2S. So when the H2S molecules acts as hydrogen donor, in that particular case, instead of a formation of oxygen, so sulfur or sulfur related compounds formation takes place or evolved. Okay, so means here in a bacteria members, if you see there no H2S will be the hydrogen donor, and if you see green plants no hydrogen donor will be water. Okay, so water is the hydrogen donor in case of green plants. So at the time oxygen is evolved. Okay, so and if you take some other kind of bacteria, so no, like for example green sulfur bacteria or purple and green sulfur bacteria point of view, what is the information we can find out here is, so here oxygen hydrogen donor is H2S, so thereby O2 is not evolved, sulfur or sulfur related compounds will be evolved or released or oxidized. Hence he referred that oxygen evolved by the green plants come from the H2O. Okay, means this particular point already we have finalized. So thereby, so what is the point we are learning now? Oxygen evolution. So oxygen evolution takes place by the green plants. So it is coming from the O2 or H2O, but not from the carbon dioxide. So thereby we are getting the confirmation. So from where oxygen is going to be evolved means the oxygen is evolved from the water but not from the carbon dioxide, and this was proved by using the radioisotopic techniques. So by using of radioisotopic techniques, so this was we have proved. Okay, we proved this one. So now according to that last and final explanation concerned, so what is the final formula or what is the final formula for process of photosynthesis means, that is 6 molecules of carbon dioxide plus 12 molecules of water in the presence of light. So it is converted into C6H12O6 plus 6 molecules of water and 6 molecules of oxygen is liberated. So these kind of reactions, so here we will be learning gradually. So thereby here what are the points now we can notice, or what are the points now we can learn here is, previously under earlier experiments point of view we have seen Joseph Priestley information, and later Johnny Ingenhouse we seen, and later Julius von Sachs we seen, later Engelmann's information we seen, later what we have seen. So in the in the 19th century, so what are the key points which are required to the process of photosynthesis we seen, and later microbiologist information Cornelius von Nile information we seen, and here so remaining information related to the green plants and the comparison between the green plants and purple sulfur bacteria, so that particular information, and finally what we have learned, so oxygen is in case of green plants, oxygen is liberated from the water but not from the carbon dioxide, and in case of microorganisms or the bacteria's point of view, hydrogen donor is H2S. In that particular case, what are the compounds are oxidized means sulfur or sulfur related compounds are oxidized we have seen. Okay, so this is some more information which we can be discussed related to the earlier experiments of photosynthesis. Okay, so next video onwards we will have the complete and detailed information, so where the process of photosynthesis takes place, and what are the sites of process of photosynthesis, exact information now we will learn gradually. Thank you and be safe at home.